So thank you all for being here for such wonderful sessions and wonderful talks. It was very engaging so far, and it's my pleasure to introduce Leyla. So Leyla is one of the graduate students here at BCS, and she works with Lethosci Lab, and I think she's very much a representative of the graduate school and the future of what's going to be in terms of the advancements in science and how we can move forward. And it's wonderful to have great students like Leyla here. So without further ado, I give her 10 minutes, and after the spotlight presentation, we're going to go to the discussion sessions. In the discussion sessions, I'll ask Mark Harnett to join us, and I'll ask Joe Monaco also to be here with us. Unfortunately, so far, I don't think Bob Desmond is going to make it today, but we'll have conversations, and we'll have an engaging conversation going back and forth over the topics that we talked about today. So let's start with Leyla, please.
Thank you. Thank you very much. Thank you for the very kind introduction. My name is Leyla Akay, and I'm a PhD student in Leyla Lab here in the Department of Brain and Cognitive Sciences. And in the Sci Lab, we're really interested in the question of how we can leverage insights that we gain from big data sets. In this example, single nuclear RNA sequencing from the human postmortem brain, to develop predictions and hypotheses that we can then test in a variety of different models, like stem cell models or mouse models. And so today, I am really excited to share a little story about how we use this approach to understand and investigate one mechanism of an Alzheimer's disease risk gene called ApoE4. So ApoE4 is one of the largest genetic risk factors for late-onset Alzheimer's disease, and it's also one of the most common across the human population. But despite decades of research into ApoE4, we still don't fully understand how it increases or modulates risk of disease. And so our question was, how does ApoE4 expression in the human brain affect the cellular and molecular pathways that can increase risk of getting disease? And so to begin to address this question, we did an experiment where we performed single nuclear RNA sequencing on the human postmortem brain, focusing on the prefrontal cortex. And we did this from a cohort of individuals who either had 1 or 2 copies of this risk gene ApoE4, or from individuals who are homozygous for ApoE3, which is a different variant of the same gene that does not increase risk of getting Alzheimer's disease. And the kind of advantage of this technique is that we're able to isolate and capture individual nuclei from the human brain and then look at the gene expression of all the genes that exist in different cells. And we can use this technique to ask over about 48 individuals which genes are more highly expressed in ApoE4 carriers, which genes are less expressed in E4 carriers, and importantly, do any of these genes that are differentially expressed between ApoE3 and 4 carriers converge onto any known biological processes or pathways? And what we found was that, indeed, ApoE4 expression was associated with significant perturbations across a really wide range of different biological processes. And these included inflammation or immune-related alterations. These included metabolic alterations across different cell types. But in particular, we were really interested in how ApoE4 expression affected lipid-associated processes. And the reason is because ApoE encodes apolipoprotein E, which is actually the major lipid transporter in the brain. So we wanted to narrow our search space into just genes that were known to be related to lipid or sterile metabolism. And so then looking just at genes and pathways that were changed by or associated with lipids, we found, kind of interestingly, that many genes involved in the synthesis of cholesterol were very highly perturbed by ApoE4 expression. And specifically, these genes were more highly expressed or upregulated in a cell type called oligodendrocytes, and that's denoted here in this column in red. So we were really kind of interested by this because the oligodendrocytes in the brain are known to be the cell types that produce myelin, which is the insulating sheath around the axons of nerves, and cholesterol is one of the major components of the myelin sheath. So what we found when we kind of looked a little bit closer into this finding was that this signature of increased gene expression by ApoE4 was dose-dependent, meaning that this increase in gene expression was higher in individuals who had 2 copies of ApoE4 relative to those who had 1 copy. And there's an interesting interaction with AD diagnostic status. So even in individuals who were homozygous for ApoE3, but who had a diagnosis of Alzheimer's disease, there was still an increase in this signature of upregulated cholesterol. And when we went into the human postmortem brain tissue and stained for cholesterol, we noticed that in the ApoE4 carriers, there was this kind of pattern of aberrant cholesterol staining around specific cells. So we next wanted to better understand why this was happening and what this meant. So we used a technique called induced pluripotent stem cells. And so what we can do is take fibroblasts or skin cells from individuals, and we can do this from people who are both homozygous either for ApoE3 or homozygous for the risk gene ApoE4. Convert these fibroblasts into induced pluripotent stem cells and use CRISPR-Cas9 editing to edit the ApoE genotype. And what this allows us to do is now to have isogenic sets of stem cell lines in which the genetic background is completely identical except for the ApoE genotype. And then we can use a combination of different growth factors to kind of take these stem cells and direct them into an oligodendrocyte-like fate. And so the video I'm showing here on the right are these cells in culture with neurons, and you can see they start to produce the myelin basic protein, which is the hallmark of myelin. So because ApoE4 is a lipid transporter, we saw the signature of cholesterol dysregulation. We next performed lipidomics on these IPS-derived oligodendrocytes. And we see that in the ApoE4 oligodendrocytes, there's this marked increase in cholesterol species, and in particular, the esterified form of cholesterol. And this was quite interesting to us because esterified cholesterol is known to be the kind of storage form of cholesterol. And if we give these cells a fluorescent analog of cholesterol and then measure its uptake and retention, we see that the cells expressing this risk gene actually hold on to cholesterol a little bit more than the neutral cells. So given that cholesterol is a major component of the myelin sheath, and given that oligodendrocytes need cholesterol to synthesize myelin, we then asked whether cholesterol accumulation in ApoE4 oligodendrocytes could affect myelination. And so we returned again to the human postmortem brain, and here we conducted what's called black gold 2 staining. And so this is a dye that selectively labels the myelin sheath. And we noticed that in the ApoE4 carriers in individuals with Alzheimer's disease, there was not only a reduction in the amount of myelin in the cortex, but also a disorganization of the sheath, as you can kind of see in the bottom row. So we wanted to kind of better understand the mechanisms of this, so we returned back to our culture system. And now we decided to co-culture our oligodendrocytes with iPS-derived neurons. And in this system, these cells will start to produce myelin. But what we noticed was that the ApoE4 oligodendrocytes in culture produce less myelin compared to ApoE3. But we wanted to understand whether this was actually like a cell autonomous defect because of the oligodendrocytes, or whether this was maybe also due to some defect in the neurons. So then we did an experiment where we switched the genotypes. So we co-cultured ApoE4 oligodendrocytes with E3 neurons, and then vice versa, E3 neurons with ApoE4 oligodendrocytes. And we saw that across different combinations, whenever the genotype of the oligodendrocytes was ApoE4, there was a decrease in myelination. So at this point, we were able to kind of describe two phenotypes of cholesterol trafficking being impaired and a decrease in myelination, both related to ApoE4 expression. But we next wanted to understand whether these two were functionally linked. So if we could rescue or alleviate this cholesterol transporting defect in oligodendrocytes, would we be able to improve myelination? And so in our culture system, we tested this idea using a compound called cyclodextrin, and this is a drug that's known to solubilize cholesterol and move it between cells. And so when we treated our iPS neuron oligodendrocyte co-cultures with cyclodextrin, we saw that in the ApoE4 oligodendrocytes, there was an increase in the amount of myelin protein being produced. Next, of course, we wanted to test this concept in vivo. And so we treated humanized ApoE4 knock-in mice. And so these are mice that are now expressing the ApoE4 risk gene. And we treated them with cyclodextrin for 2 months. And we assessed their myelination using transmission electron microscopy. And we noticed an increase in the myelin sheath. And we also wanted to understand how this could impact cognition. So we did a behavior experiment called the puzzle box, where we present the animal with a difficult task, and we measure its performance over a couple days. And we saw that while the non-treated ApoE4 animals did not have much of an ability to learn or synthesize new information, the treated animals showed an effect. So given all of these questions. I think like one kind of question that stood out to us was, okay, this is interesting, but like what does this have to do with Alzheimer's disease? And so I think the big picture is like how does oligodendrocyte dysfunction or function impact neuronal activity in a way that could kind of lead to mechanisms of not only memory function, but also memory dysfunction. And so moving forward, we're really interested in looking more deeply at the kind of relationships between these oligodendrocytes, the myelinating cells, and also neural activity. And so we're kind of playing around with transplanting these iPS-derived oligodendrocytes into more complex three-dimensional models of neural activity. And so what we've been doing is using cortical organoids and taking these iPS oligodendrocytes, shown here in green, and transplanting them into organoids. And we can manipulate neural activity in these organoids by expressing channel rhodopsin and then fuse them to another organoid in which we can measure calcium activity using GCAMP, and this creates a little assembloid. So we can both monitor activity as we simultaneously drive it, and then ask how do oligodendrocytes affect these kind of complex neural functions and ensembles. And with that, I would like to thank you and thank my mentor, Leyla Akay, and everyone who kind of helped and worked on this project. Thank you.
Do young people with ApoE4 show any issues related to myelinations that are consistent with the results you're seeing?
Very interestingly, there was an fMRI study that looked at white matter volume in infants with ApoE3 and 4. And what they found was that it's not that ApoE4 necessarily has like a global decrease in myelin, but there were differences in white matter composition across different brain areas. So in some brain areas, ApoE4 carriers had actually increased myelin, and in other areas, they had decreased myelin relative to E3. So I don't think that it's fair to assume maybe that there's like an overall broad inherent defect in myelination caused by ApoE4, but maybe some kind of age or stress-related phenotype.
Ken, please go ahead.
Would you expect to see deficits in nerve conduction if you look in, say, the periphery as a more remote possibility, but even centrally with devoted potentials, like you detect some slowly?
Yes, absolutely. We're very interested in that, like how the insulative properties of myelin, if it's decreased in E4, could affect actually circuit function. And yes, we haven't looked at peripheral myelin, so I don't know the answer to that, but I think that would be a very interesting possibility to explore, for sure.
I also have a question. So when you do transplant this into this sort of like organoids, how do you ensure that, like, this set of organoids that you use and reuse, for example, from different growth of this organoids, are they going to be similar in terms of their structure or not? Because each of them kind of like grows a little bit like, randomly, right? Mm-hmm. So how do you control that, and how do you take that into account? Like if you want to create a system that is reliable, basically, that each time you're experimenting, you're dealing with almost like similar type of structure, at least to begin with?
Right. I imagine that you could screen these assembloids for making sure that they have the right kind of cortical architecture, they're expressing what you would kind of hope for them to express in terms of neural activity as a response. But that's definitely a good question. I think something that the field is really trying to work on. Yeah. Yeah, so our hope is that it is. All of the data, like the kind of counts and reads from the RNA sequencing, is available on Synapse. And we have, Juna is another grad student. She did all the computational work. Her GitHub has like line by line how to like remake all of the plots in our paper. But our hope is that this is able to be accessible and used and reused by people. That's something we're super interested in, and also kind of looking at the impact of this gene expression on earlier ages as well. We don't have that data right now, but I think that would make for a very interesting story, definitely.
Okay, let's thank Leyla one more time. Thank you for your nice talk.